Good evening, everybody. Welcome to Igniting Leadership, our second in a series that the Ignite Women's Healthcare Group is putting on. I'm really excited. My name is Susan Feigen Harris, and I'm here with Dr. Maria Elena Bottazzi to talk about the state of COVID vaccines. So before we jump in, Maria Elena, I'm going to brag on you for a little bit, if that's okay. Dr. Bottazzi is the Associate Dean of the National School of Tropical Medicine here in Houston at Baylor College of Medicine, and she's the co-director of Texas Children's Hospital Center for Vaccine Development. She's an internationally recognized vaccinologist and a global health advocate for neglected tropical diseases. And she is my friend whom I greatly admire. She's a leader in science and now really is at the heart of the race to produce a vaccine that we all need to combat COVID-19. And her work is directed at producing a vaccine that is affordable and effective, not just for us, but for other countries as well. So we're going to talk a little bit tonight. Hopefully we will go through enough of the issues that we will answer many questions, but I know you're getting information about asking questions, so please feel free to do that. And at the end, we should leave about 10 to 15 minutes for a question and answer. We'll try to make sure we touch all of it. So I'm going to start, Maria Elena, with really, for me, what I think is sort of the core high-level issue with respect to vaccines. So there, in my view, I guess, thankfully, are so many vaccines that it's really hard for the public to discern the difference. There are RNA, DNA vaccines. There's a protein-based vaccine. There we hear about these viral vector vaccines that use whole inactivated viruses. I feel like we as a global community are learning probably more about immunology and vaccinology and production and manufacturing than any of us thought we would ever learn. Can you help us demystify the variety of vaccines and the categories and help us so that we can keep these straight? And then we'll get into the next. But, you know, the first ones to come out, the second ones to come out. Tell us what's happening.
Well, first of all, Sue, again, a pleasure to just be in this conversation with you, because as you said that you're my biggest fan, I'm your biggest fan. So, you know,
so we're the mutual admiration.
Mutual admiration here. Second, of course, thanking the organization, thanking, you know, the Igniting Leadership. I'm a big proponent, certainly, of women leadership. So I'm just delighted just to be sitting and probably, you know, with an audience of bright innovators. So it's a pleasure. And indeed, you're right. I think the good news is that vaccines, I hope, everybody now understands that they are definitely probably the best public health intervention that we have ever, you know, as humans were able to not only discover, because it has made some amazing savings around the world for many diseases, right? So I don't think I need to remind everybody about that. But you're right. I think that the categories is a concept that it's from that first pillar, right? So how do we produce vaccines? How do we make them, right? What's the prototypes of these vaccines? And more than anything it's kind of like, what's the machinery that produces the what we want our body to get exposed? And so I think when we say about machinery, that's where the concept of these categories come through. So at the end of the day, what our body needs to see, it's ultimately a protein. And the protein then gets processed by our cells and our body, and then it gets presented, and that's what we respond against, right? So if you present the entire virus, whether it's inactivated or whether it's, you know, manipulated, because of course we want to inject something that is not going to cause disease, the body needs to chop it all up and take all the different proteins, and eventually you present what you really think that it's important. If you take an actual protein directly, meaning the protein-based vaccines, you're just asking your body to do minimal work, which means process such a protein and present it. If you take an RNA or a DNA, you're actually first asking your body to make them into protein. So the genetic code becomes DNA becomes an RNA molecule. The RNA molecule has to become a protein, and then that protein needs to be processed. And the viral vectors is that you're mocking a virus. You're just swapping genetic codes, similar to indeed the whole virus vaccines, except that your backbone of the virus is an unrelated, totally irrelevant virus. Instead of taking the COVID vaccine virus, the COVID virus, you're taking an adenovirus, and you're just swapping genetic codes.
So that's sort of, those are like the immunology uses that we've seen with cancer, et cetera. That's sort of the great. So one of the questions I have now to sort of take that a little further before we go on is, so of the Operation Warp Speed vaccines that we know about, can you slot them into these categories? Which one's an RNA, which one's a DNA, right, which one's an adeno? Yeah, I mean, can you just help us understand which one is using which technology so we get a baseline sense before we go forward?
Sure, and I also want to make it clear that all the licensed vaccines that currently occur today, meaning that we already use widely, only are from two categories. The live virus vaccines, whether they're inactivated or attenuated, or the recombinant protein vaccines. Those are the only two platforms that we know we already have licensed vaccines for.
So what you're saying is the vaccines that are being produced by Operation Warp Speed, unless you criticize me on this, are not being produced with a vaccine platform that has ever been used before for purposes of a vaccine.
Correct, meaning they have not been widely used in population. So for example, Moderna and Pfizer BioNTech, they use an RNA platform. We've never had a vaccine license using RNAs.
Okay.
In fact, RNA, the concept of using RNA have been developed originally for gene therapies, for cancer therapies. Right. And they're now kind of repurposing the original intent to use them now as a preventive vaccine strategy. Then you have the vaccines such as AstraZeneca with Oxford, which is interestingly, it's an academic pharmaceutical collaboration.
Right.
Uses an adenovirus. That's one of those viral vector vaccines. We have tried years and years to use adenoviruses as the viral platform, and we still don't have any vaccines that have led to being approved. But those were originally intended to become vaccines, not like the RNA or even the DNA that were mostly directed towards therapies, gene therapies. We also have the only protein-based vaccines are the Novavax vaccine, which I don't think you've heard it maybe too much. It is part of the Operation Warp Speed. Yes. But it's a little lagging, a little bit behind, but it's actually probably trending very positively. In fact, we think that it's one of those who will eventually show a lot of effectivity and because it uses interesting recombinant platform. It's a novel technology, but it's still in the spirit of recombinant protein. And then the only one that really uses a mostly bread and butter within the Operation Warp Speed is the Sanofi collaboration with GSK, who uses a recombinant using a production system that uses insect cells. We can go into a lot of how do you produce vaccines too. But the Operation Warp Speed are two RNA technologies We have, the viral vector, which is the AstraZeneca, which are the ones that are the most advanced: Moderna, AstraZeneca, and Pfizer. And then there's others coming behind that uses either other viral vectors, like J&J, Johnson & Johnson is a viral vector. Novavax is a recombinant protein. Sanofi is a recombinant protein.
Okay, so we've sort of established, thank you for doing that, because it's very confusing, and I appreciate that. So now, we can't go on, I think, without talking about yesterday. There was a big publication, and I think all of us were initially really excited about it. And then this morning, you see the criticisms of it, but Pfizer issued, I guess, a press release with respect to what seems like really optimistic news. They had a trial with 94 participants. I thought it was 94 participants, or the results were based on 94 participants. Maybe you can help me with this. Mm-hmm. And it sounded as though they received their first dose just before the election. I noticed that. So it was very recent, and there's new data, but it sounds as though there's significant coverage and viral response or immuno response with that. Can you tell us a little bit about the Pfizer vaccine? And it also wasn't part of Operation Warp Speed. So, should we be hopeful? Should we have healthy skepticism? What does that mean in the life of all these vaccines and what we can expect?
Mm-hmm. Well, first, let me clarify a couple of things. So irregardless of whether it's within Operation Warp Speed or that will eventually join the Operation Warp Speed, Operation Warp Speed was really a vehicle of a couple of things. One, of course, funding, right? Mm-hmm. There's... Right. The U.S. government amazingly have invested billions of dollars. Even if you see it in context with the rest of the world, there's a lot of distinction between how much money the U.S. has invested versus the rest of the world has invested. We can go into that maybe another day. The second is that the way that these companies advance their products, like Pfizer or again J&J or whoever, they had to go through different stages of clinical trials, right? But mm-hmm. when they reached the phase 3, that's where the harmonization and the hardcore dollars from Operation Warp Speed was invested to try to ensure that there was an understanding of how those blueprints or protocols were going to be done. And in fact, if you even, look deep, you actually can search and you can see the published protocol or blueprint that Pfizer was going to use. So the way that they designed these studies, and I know it's complicated. So first of all, they're large studies, multi-center. So for example, even in Houston, through the University of Texas, they were one of the sites that tested the Pfizer vaccine, where I believe their allotment was 500 individuals. For example, Baylor College of Medicine participated in the Moderna vaccine. So, we, for example, from this press release, we do not know from which center these so-called events happen. The second piece is what I just alluded is the whole vaccine trial was more than 44,000 people, right? And from there, half gets the placebo, half gets the vaccine, which you don't know which half, right? So for example, if UTMB enrolled 500, probably half were placebo, half got the vaccine. And then the way that they do 2 doses, right? 1 month apart. And then the FDA require them that they have to, at least for them to look at the data, follow them for at least a couple of months. So that
how is it that they were able to come out with data so quickly?
Because,
or should we be skeptical of the data that came out?
No, you don't have to be skeptical, but you have to then understand that that press release was a little peak of interim data, right? Okay. So it's just like, let me actually open the oven and see if the cake is sort of done. And then you close it and you have to still wait for the cake be baked, right? So that peaking of the data and can have positive outcomes because they already can get a little hint. But statistically, they may get penalized because more you look at the oven, more likelihood your cake is not gonna raise the whole, right?
Great analogy. Great analogy. Good analogy.
So sometimes, and in fact, Pfizer originally in their protocol said they were going to peek at the data 4 times.
I see.
But then they, in consultation with the FDA, they kind of deviated. And in fact, now they're not going to look at the data 4 times. They're all probably going to look at the data only once and then wait at the end, because there's penalties statistically, right? In biology of how you peak, because you have to unblind and see some things. The second part is the so-called 94 or 90% efficacy they saw. It's on the basis of events, meaning.
Oh, I got the number wrong.
Right? So it's not participant, it's
gotcha.
Amongst the people they vaccinated, how many
of the 44,000, they had 94% effectiveness. Thank you for correcting.
They had 90% efficacy, but it was because they saw 94 individuals who had an event. And by event, that's what I don't think we also understood from the press release, because an event could be that these 94 people had not very severe disease compared to a group that actually had very severe disease. And therefore, what you're seeing is that your vaccine had an impact from reducing the severity of the disease.
I see. So what you're saying is we don't even know the acuity of the individuals that ended up.
We do not know what demographics were those 94 people. We do not know what age those 94 people were. We do not know how was it that they were protected from severe and became less severe, or were they protected from less severe to asymptomatic? So there's a grading scale. We don't also know nothing about of course, how much immunological response they got. We don't know how long-term durability this response will have. Again, it's optimistic because when you peek at the data and you see that your cake is actually baking and it's kind of looking nice, you hope that at the end it will actually not only look nice, but it will taste nice. So now we just have to wait for the whole thing to end.
Well, what you just did is the next question I was going to ask you, and that is sort of tell us as a vaccinologist, I mean, all the issues that someone has to think about as you create a vaccine. So now tell us, because you and your partner, Peter Hotez, have a vaccine. You had a vaccine that was frozen. I mean, tell us that story. And then tell us about this concept, which I don't think I appreciated until I started talking to you about all of this. The approach to the development of the vaccine through this, it's called the PDP model, but this product developed partnership, because it also then allows us to talk about sort of the international component. So can you sort of, take us through that story a little bit?
Yes, sure. And so I just want to, of course, clarify that because we are working under an academic, I guess, premise, because we are scientists and our organizations by nature have a nonprofit mission, right, to serve our communities, whether it's locally or globally. Our drive, with me and Peter and our vaccine center, is that all the vaccines we attempt to discover and advance should, by premise, be accessible to anyone. And so when we work in these partnerships that you mentioned, we tend to always find partnerships that maintain that philosophy and that premise that we, of course, people have to make money at some level because you have to invest, and of course they have to recover the investment. We believe that certainly in a setting of pandemic, the drive should not be making money, right? The drive should be make the vaccines in a way that eventually can be deployed to whether you are in a high-income country or a low-income country, whether you can afford it or you cannot, right? So our story primarily is we've been working in coronavirus vaccines for the last 10 years. with this spirit that they have to be accessible, safe, effective, but at the same time affordable, we always tended to think that novel technologies, like for example, this Pfizer technology, these RNA technologies, you know, it's like, again, another analogy. When you're going to buy the latest iPhone, it's going to cost you $500. If you were to buy a prior, you know, version, or maybe something that already has been in the market for a while, you know, the prices, you know, get lower. Why? Because there's more people that can produce the iPhones. The parts are cheaper because they become generic, you know, so you share the procedure. So working with vaccine platforms that we already discussed were already used in others allows you to know that there are many producers that can replicate the process, that the reagents that we use are not proprietary and not expensive. And that already facilitates the vaccines to be of low cost. So for example, the vaccines we work with, and the one even for COVID-19, our hope is that it won't cost more than $3-4, maybe $3 a dose. These RNA technologies, these DNA technologies, even the adenovirus technologies, I understand that even with like not at cost or even with some losing cost economics, they're still quite expensive, maybe $7-10 a dose. And you may not think that $10 is a lot of money, but you know, 2 doses is $20. And then in a country like Honduras that you need to vaccinate, I don't know, 7 million people, you know, count the amount of millions of dollars. And albeit we even have barely, you know, public health, you know, essential medicines, is not going to be very easy, right? So that is why the distinction from our platform is we prefer to use already bread and butter technologies that we can know much better how safe they are because they've been used before. We can reproduce them and scale them because it's been done before. We already know the partners that already have found that balance of cost versus, of course, return on investment. And that's how we now, for example, our recombinant protein vaccine was transferred over to not necessarily a big multinational pharmaceutical company, but a type of pharmaceutical company that it's called a developing country vaccine manufacturer, meaning, and arguably, I don't want to disparage the fact that just because they are developing country vaccine manufacturers that are less than a Pfizer or less than an AstraZeneca, they equally, quality-wise, they have to be equal, right? You know,
Right.
you know, there's no difference on something made by Pfizer or something made by an Indian company because it's very rigorous. But because they are within a country that themselves struggle with the fact that they have a lot of poverty and that they need to produce their products with a hybrid of serving for the public health, because probably in India, there's not, you know, third-party payers through, you know, insurance companies like we have in high-income countries. They therefore have to provide vaccines at very low cost and it's a mass volume business, right? So that's the real distinction.
So you sort of lopped over the fact that the company that you have partnered with is this BioE, which is based in India, correct?
Correct.
And can you tell us a little bit? So you've got the protein-based technology, which has been used, if I'm correct, on multiple occasions for other vaccines. Like, did I get it right? It was like, is it a hepatitis vaccine?
Hepatitis B vaccine, human papillomavirus vaccine.
Okay, so it's very well documented in terms of its efficacy. And I gather you believe that because of that, the viral response will be known, or you're hoping it'll be known and higher or high in terms of making sure of its effectiveness. And now you're looking at all of that plus the safety, and it's all being done in India. Is that correct?
Right. The first strategy. So we transitioned the what we call our know-how And we actually, engineered a prototype, which is this recombinant protein from the COVID virus
In the lab here in Texas.
in the lab here in Texas. So
At
our
Texas
lab,
Children's, yep.
correct. Our lab really, what it does is actually does make real prototype vaccine products. We also do some of the evaluation, albeit not in humans. We do them, of course, in the laboratory models. We then Biological E, which is amongst these developing country vaccine manufacturers. And in fact, Biological E is probably one of the largest vaccine producers globally. And I know we're going to talk about this a little bit later, but we've never talked about vaccines with regards to where they come from and who makes them.
Well, we should talk about it now. I mean, we've got this whole concept of, I mean, we don't have to go in order.
Right.
We were talking about it before, this whole concept of vaccine nationalism. And
Right.
I mean, that's a new thing, right? I mean, we should talk about it now. Tell us about the international component of this, because I think only now are we becoming sensitive to it.
Right, because to be honest, I mean, it doesn't really matter whether it's a Chinese vaccine, a Russian vaccine, an Indian vaccine, a US-based vaccine. In fact, Pfizer probably most of the times don't necessarily only manufactures in the US, right? In fact, Pfizer probably manufactures in Europe. And to be quite honest, for example, AstraZeneca has entered into an agreement where Serum Institute of India is the one who's producing for AstraZeneca. So we've never categorized vaccines from the source of where the manufacturing facility is located or which country is the one who is advancing the discovery, right? Because ultimately they all have to pass through the same high bar quality. The regulatory bodies, of course, have to, they probably do a lot of audits of all these facilities. So I can assure you that most of the vaccines that we already get most likely already come from either an Indian manufacturer or potentially, a consortium of manufacturers, because as you know, vaccines require, a whole, group of manufacturing facilities and factories. And for example, Serum, who is the Indian, one of the largest Indian manufacturers, also have factories in Europe. But at the end of the day, it's a Serum Institute vaccine, right? So it goes both ways. So I think that we have to forget about, oh, I'm not going to use this vaccine because it's the AstraZeneca made in Brazil, or it's the AstraZeneca made in India. Because at the end of the day, who regulates looks at the vaccine as independently of where it comes from, because it's all regulated.
So, one of the other things that I thought of as you were talking that we probably should address is the issue of not just the manufacturing, but then the distribution, because at least, and I can't remember which of these vaccines has to be stored at minus whatever degrees, and just the whole concept of how you would ever produce and then distribute it. One of the other benefits, that's an issue, so I want you to talk about it, but it seems like one of the benefits to having sort of a tried and true bread and butter concept is that the distribution methodology will not require some crazy refrigerators or something that have to be used. So maybe you can talk about all that, because I'm curious about it.
Absolutely. And I want to, however, put it in perspective. We've never distributed anything, medicines or vaccines, in the context of a global pandemic. I think this is really the first ever, right? Right. One thing is that we, of course, have done this in scenarios of outbreaks. We may have even done some of these in the scenarios of epidemics. And the moment this became such a global pandemic, I think no one ever had, even those who plan for pandemic preparedness or epidemic preparedness, I don't think their blueprints or their guides took into consideration such, a global, the fact that we are going to have to distribute this to absolutely every single last person that lives in the face of this world, right? Ideally, from pediatrics all the way to elderly. No vaccines generally are to be deployed into the entire demographics, right? So pediatric vaccines tend to be for pediatrics, And therefore, the way that they plan their production, they plan their distribution is on the basis of new birth cohorts, right? How many children get born, how many children, right? So then you have the vaccines that we elderly have to start using, like, like the Shingrix or the, pneumonia or whatnot. But again, we can manage that because you know that you need to produce a reasonable amount, but not, an enormous amount. So that's already a challenge, right? Even for bread and butter vaccines, even as ours, where instead of making hundreds of millions, we need to make billions, right?
Well, and I think the issue you and I haven't really talked a lot about is this issue you're raising now about children, because of course you and I are both have spent a long time focused on pediatrics and kids, and we know that, even whether it's medical devices or vaccines, I mean, they're usually, it takes a long time to do something in the pediatric world, and sometimes it's being used off label. So I think as an adjunct to what we're talking about, and as we talk about even the safety and effectiveness, efficacy for adults, the question will be, what about kids? How do we even begin to test that? And I don't know if you can talk about that now, but certainly I think we need to keep that in mind as you've just identified.
Right. And indeed, as a sidebar, we need to recognize that all the studies that are ongoing, they do not go down to the point where they're being evaluated in the real pediatric population, meaning
Right.
infant, maybe, I think that the lower age range that it has been evaluated is probably the teenage range, right? The 12, 13. So any kid below 12 years of age, most likely in this first tranche of data, we won't have any data because it's not been, there's no trials for the pediatric population
So
yet.
would you conclude? So my next question really had to do, I sort of told you, we get back to talking about our bodies and how a vaccine works in terms of creating virus neutralizing antibodies. So let's talk about that both in the adult and the pediatric population, because it begs the question whether or not a child should get this at the end of all this. I mean, I know adults are asking, how do we know about the safety and efficacy of these vaccines? So maybe you can help us differentiate between how you, as someone who creates vaccines, sort of looks at the safety and efficacy of the vaccine, and then how do we look at this in terms of these first vaccines that are going to come out that are novel, that use these novel platforms? Because yours, I mean, you've already said it's not on this warp speed the way the rest of them are. So maybe help us a little bit with that, because that's got to be a common question.
Well, I think first of all is to remind everyone that, even after the fact that, these clinical trials are completed, the premise of what we call post-marketing or post-licensure surveillance continues forever. Meaning even today, even for vaccines that we've always used in our kids, right? There's these systems that continuously safety data is added to these databases, right? So that we can evaluate more and more not only their efficacy. So efficacy is what you look at when it's under clinical trial studies, and then effectiveness is evaluated once it's deployed in the community, and you continue building all these safety assessments.
So do you mean with efficacy, is that how it creates the virus neutralizing antibody, and then effectiveness is sort of when it gets to herd immunity? I mean, help us sort of.
Yes. So when we talk about efficacy is under a control study where you're actually keeping track of, indeed all these, not only neutralizing antibodies. Of course, we also look at cellular responses. We look at, blood chemistries. We look at even site of injection, so there's, it's a whole ecosystem of clinical things that the physician looks like, but it's under a physician purview because you are part of this study, right?
Right.
When the vaccines get approved and then they get deployed by your healthcare provider or your pediatrician or your whoever doctor, or even when you go to CVS, right? Then
Right.
it's the fact that it's now not under a study, but
Mhm.
but we as individuals who use it, if we complain and if we of course call our doctor and say, oh my God, I got this fever because I got the flu vaccine, all those data eventually do get reported. And
Mhm.
all that data eventually accumulates, like you said, how many people are getting vaccinated, that will lead into the coverages, how many people get actually vaccinated, because herd immunity is directly linked with usage, right, of coverage of use. More people uses it means that more people are going to be protected, whatever percent protection it may be. And therefore, when it's like you putting yourself inside I don't know, a soccer field with all the players, right? If only one person is vaccinated and the rest are not, there's more likelihood that there's more infection. But
Right.
if you have most of the
Including from the person who's vaccinated, correct? I mean, the vaccination, and it's at least my understanding, and this is, a layperson's understanding of herd immunity, is that if you do not have enough of the herd vaccinated, you yourself can,
also get infected.
right, you need the other people around you
Correct,
to
because
also be vaccinated.
correct, because if a vaccine is not 100%, and of course, as you know, there's nothing ever absolute, right? In science,
Right.
there's nothing absolute.
Well, especially with this, right?
Right.
Initially, okay.
Right. So if I get a vaccine, but I know I am 50% protected or 80% protected or 90% protected, there's always that 10% chance. But if I'm surrounded by others that are also vaccinated, there's a less likelihood that I am going to get infected because I'm in a bubble, right?
Right.
But if I'm surrounded with a lot of people that are still transmitting and are still, of course, not protected, I have a probably higher chance that, God forbid, I may be 90% protected, but I also have that 10% that I may not, right?
So we didn't discuss this last week when we were sort of talking about this, but it sort of begs the question about the whole concept of herd immunity now, because I've always talked about or thought about herd immunity in the context of vaccination as opposed to in the context of massive exposure to disease, and hopefully everyone gets sick and you create an immune response. We have no data on that. Is that correct?
Well, I think that the problem is, again, I think it's a Russian roulette game, right? Because if we knew that that you would get seriously sick and that I wouldn't, therefore I would say I can go out and infect myself and I don't care. But the problem is I do not know that. So by you letting this rogue and having this concept of we should just let ourselves randomly see what happens, unfortunately this is why we have seen so many deaths, right?
Yeah.
Because it's a spectrum of a disease, unfortunately, and we don't really even know why you do get severe disease and eventually die and why I do not. Yes, it's a combination of maybe because my genetic background is favorable. What that genetic background is, I still don't know. Maybe it's because indeed I am more healthy than you are and I have less risk factors. But what exactly beyond understanding that, of course, if you have diabetes, if you're overweight, if you have cardiac disease, of course, all those are, common sense factors that are always going to burden you, even for other diseases. We don't even really now know whether, if you're a kid, you have less chance of getting severe. Now we actually don't even really 100% can assure that, right?
Right, because kids are getting sick.
Because kids are getting sick. So since we don't know, we have a lot of known unknowns, we cannot just say, look, just venture, and if you happen to get severe disease, you're at your own fate. I think that's unethical, immoral, and I think that we should absolutely not do that.
Thank you for answering that, because I think it's an important question, and I like people to hear it as often as they can, because it's it sort of shocked me, personally, I will say. So begs the next question, which is, and I know Peter gets asked this question all the time, would you take the vaccine, the first vaccine produced? And I think that's what everyone's asking, you know.
So process-wise, I want to make sure everybody understands that it's very rigorous, and which means that if the agencies, and it's a combination of agencies, so it's the US FDA in the case of the US for regulatory, there's all these safety monitoring boards, there's the VRBPAC, which, you know, and then eventually there's those agencies that confirm who would be the appropriate target population that can get it.
Right.
So if you see that under an emergency authorization use, most likely it means that it's close enough that it could eventually reach licensure, but that because it's an emergency, they want to roll it out earlier. So if indeed that happens, and it just magically happens that I fit within that category of the initial target population that I can qualify for it, I would say go get it, because it's already going to show sufficient safety that, you know, that merits using it. We already, albeit we need to be clear with the population of what's this efficacy percent, so that people don't get blindsided saying, oh yeah, even if it's 90%, oh, then that means I'm not going to use a mask and I'm going to go off and party.
Well, that's an important, right? You just raised two really important things, Marielena. The first is this concept that just because a vaccine is ready, everyone doesn't get to get it, right? It's that there's a limit. I mean, you've raised it a couple of times. There's a limit on how much will be produced, and there's going to be a priority, hopefully, in terms of who should get it first. And so what you're saying is, you know, average Joe probably will not be in the first batch of people that would be receiving the vaccine. And that's an important component. And then the second thing has to do with the transparency in terms of, you know, what led them to ensure that this was safe and effective for use in this population.
And including the selection of why the initial first targeted population was selected, right? You know, which we are hearing probably healthcare workers, you know, and service people, like, you know, our policemen, our, you know, the people who really do a lot of community service, you know, in our communities. And then, of course, there's also the logistics, because, for example, if you take the Pfizer example, I think right now they said that for the first launch, they probably can only provide, you know, I don't know, 20 million, maybe 50 million. And on top of it, this is one of those that have to be kept at sub-freezing temperatures. Yeah. So the logistics will be a little bit complex. But let's say they even go, you know, through that hump, and we have these 50 million doses. We're 350 million, I think, plus people in the US. How are we ensuring that every state, every city gets an amount? And I think that the premise of how this is being rationalized, and in the US, I think the framework was designed by a commission from the National Academy of Medicine. So the framework of how the equitable distribution, at least for the US, is gonna use that guiding principle. In the rest of the world, they're looking at the World Health Organization framework, but it's very harmonized. And I think at the beginning is equality in the distribution. So let's say you get, if 50 million means, I don't know, 20% of the population, whatever that number is, I'm not good in math, that it's equally distributed. But then the next tranches are not on the basis of population density, but it's probably on the basis of where do we see the most transmission, what the epidemiological, right? And as we know now, there's states that are controlling it much better than other states, for example. There's countries that are controlling it better than other countries. So it's complicated, but at the end of the day, the aspiration is, even though we first start with small numbers and with very targeted, of course, on the basis of how uh, efficacious and how safe is in that population. You know, if you fall into that category, go get it, because, you know, certainly it's not gonna, you know, do anything, no harm. In fact, it has a much more benefit than the risk. If it's not the pitch perfect vaccine, if others will come out later, which probably they will, that's why some of us, including, for example, ours, even though we're aspiring to first advance it in India, eventually open it to the globe, and maybe even come back to the US. These vaccines can either replace, they could boost and complement. So there's nothing wrong with, if you got one, eventually you may get another one, even though that's a logistic that also needs to have very special tracking, right? You want to know what you got first, so that you don't kind of do a little bit of too much of a mixture.
There are two questions. Don't let me forget. One is about the masks. But before I ask you about the masks, you know, what you're raising is what we all go through now with vaccines anyway, but it sounds like we won't know. And everyone, we forget that you learn about vaccines, and there are different, you know, updates with and versions of vaccines that we've lived with over the years, as they've been improved upon. And what you're saying is that's what's going to happen with this vaccine as well. And that, you know, there are all kinds of issues I'm thinking about as you're talking about it. And that is, you're talking about the dosing schedule, boosters, you know, how often and when you should get a booster, how long the immunity lasts. I mean, all of that, I guess, even when we have a vaccine, is yet to be determined. And so what I'm realizing is that this is sort of this major long-term scientific and data collection process that we're going to have to incorporate into everything we've been doing for vaccines that we've lived with for many years. This is probably the first time many of us have addressed an issue that we're going to be living with. You know, we all have gotten the polio vaccine, and you know, all these other vaccines. We didn't live with the existence of all of the kind of data collection you're now talking about. That's going to have to go on for years to come.
And also remember that I know it's been an eternity, but it's only been 11 months, right?
Oh, I mean, it's amazing.
So it's amazing in itself. I have to tell you personally, I can assert that it's been quite intense and
I'm sure.
and nerve-wracking, because you want to even do it faster, but you have to, of course, understand that there are things that you cannot, because you have to wait for the data, you have to wait for the safety, you have to wait for the, even in the area of manufacturing, and I think at the end we probably hopefully won't go back to that it takes 10-20 years to develop a vaccine. Maybe we can, you know, get a little bit better efficiency,
Right.
but I don't think it's sustainable that vaccines get developed in 11 months, right, or 12 months. I think we're gonna have to find where, sure, maybe not 10-20, maybe 3-5, but not certainly 0-1, right?
Right.
And I think people, you know, and I know that people are anxious, because indeed, if we add to the rest of the technologies, like, you know, wearing masks, physical distancing, contact tracing, diagnostics, right? There's, you know, in the technology world, even using, you know, cell phone, or even telemedicine, you know, all these technology are not going to go back to the normal business as usual way of doing things before, because we've learned and we've adapted. And I think hopefully this will actually bring a better way of doing things, with better investments and with better solutions for all the global goals that we have. But I don't think that we're going to be able to sustain this, you know, absolutely, you know,